tools today. So the main goal is to get acquainted with different kinds of tools as we've been doing for the last class. Please go, there are three specific websites that I would like you to go to. First website is the EBI tools for MSA, which is multiple sequence alignment. The next website is some details about one of these tools, one of family of tools called T-Coffee. So if you remember, we discussed two kinds of multiple sequence alignment algorithms, which are actually used. One was iterative and the other one was progressive. So we will basically, T-Coffee is one of the versions. And then after you have gotten the alignments, the next thing to do is to actually use what's called a multiple sequence alignment viewer, which allows you to highlight certain properties of the alignments and basically understand more about what you've obtained. And here there is a website for the tutorial, which actually shows you how to download. It gives you an alignment and it shows you how to basically look for specific things in that alignment. All right. So going first to the website of this EBI tools for MSA. This is how the website looks like. So it gives you some information. We have already looked at the emboss tools from EBI, which were for pairwise alignment. This one is for multiple sequence alignment. So you have multiple tools here and it gives you a one-line description of what these tools are doing. So you have Cluster Omega, which is a tool that uses seeded guide trees and hidden Markov model profile alignment techniques. So seeded, whenever you see the word seeded, it essentially means that you are using um, the idea that whenever you have exactly same k-mer showing up in multiple sequences, that exactly same k-mer might be. It's not always 100% true, but it might be the site of starting a multiple sequence alignment. And these kinds of algorithms I haven't discussed. These are called branch and bound algorithms. So the idea is that you have start two alignments, one alignment on the left side of the seed location and one another alignment on the right side of the seed location. And typically the alignment almost goes towards linear time for pairwise and n squared times for multiple pairs. And hidden Markov model essentially is a way in which you can store information of a profile. We all know how a profile looks like. Hidden Markov model captures a little bit more information than a simple profile, which is capturing just probabilities. So if you are just capturing probabilities of a nucleotide or an amino acid at a specific location in your profile, that's what's called the probabilities represent a hidden Markov model where the chain length is 0. So it's a Markov model with chain length 0. Hidden Markov model has some other information that it stores. It's beyond the scope of this course currently, so I would not go into it. But the basic idea is that Cluster Omega is using seeded guide trees and it's using profile alignment techniques, which I've already discussed in class. Then you have something called emboss cons. So it creates a
Sir,
consensus sequence. Yes.
sir, you just mentioned that cluster omega is used for proteins and amino acids and nucleotides also. Sir, in class, perhaps we discussed that hidden Markov model, that Markov model is used for nucleotide, I guess. Sir.
So Markov model is just a way of storing information about probabilities. All right. It's not restricted to only amino acids or nucleic acid. It can be done for even, say, predicting if tomorrow is going to be rainy day or sunny day. This is what you would probably, if you would look at some tutorials online, this is how they start to talk about. So hidden Markov models or Markov models, they are
Okay, sir.
essentially a way of storing probabilistic information of multiple series of events.
Okay,
Okay.
sir.
So emboss cons creates a consensus sequence from a protein or nucleotide alignment, right? So for example, if you have a very long alignment, many nucleotides, instead of doing it by hand, you can just feed that alignment into emboss cons. So, I'll just click into each one of these. So let's say we go to cluster omega, right? So you can choose what kind of alignment you are having. So you are having set of proteins, DNA or RNA, right? You can also look at the format. You can click here and understand what kind of format they allow you to have sequences in. You can, for example, put an example sequence. So here we have put example sequence, 3 example sequences, right? The output format can be a cluster W format with character counts. It can be a Pearson FASTA file. So please look into exactly how each one of these outputs look like, right? And then you can have more options, which essentially tell you something about the clustering itself, so the alignment itself. So you just play around with these tools to see how they affect the alignment. So you can either have aligned order or you can have input order. So input order would basically be showing you exactly alignment in the same order of your sequences. The aligned order is going to show more similar sequences on top and least similar sequences at the bottom. Uh, you have a number of iterations because, as I said, it uses hidden Markov model to make profiles. These profiles are typically simulated, so you can actually have more number of iterations. The more number of iterations you have, the more confident you'll be about your results. Uh, I think in all the cases where you have iterations, if you choose the higher number of iterations, it's going to take longer time and it's going to give you a much more precise result. So as we do for anything, any of these tools, we'll just submit the job, and at the end, I think you shall be quickly looking at the results because this was the example sequences. So this is how you see. So you have mouse, human, and cap5. You have these 3 sequences, right? And again, as we saw in class, wherever you have all the letters same, you have a star. Wherever you have similar kind of amino acids, you have this conservative alignment, you have 2 dots. You'll have a single dot where it's semi-conservative alignment, and you have no dots where there is no alignment, right? So this is just the basic alignment. You can look at the result of this alignment. So each of these are individual text files that you can click and you can see what these text files are. Then you have from these sequences, you can get some kind of a tree. You know what these trees represent. These trees are basically representing distances between these samples. What are these branch lengths? We'll talk more about it during the phylogenetics lecture. You can see that you can even get like a phylogenetic tree. So this is basically using another method called neighborhood joining tree. This is the guide tree, which was used initially to make the results, to make the alignments. And then you can have different versions of this viewer. So essentially, if you want to put the results of some alignment in a manuscript or in a paper that you would want to publish, or have a website associated with it, like an online link, this allows you to create something like that. And these are the submission details. So this is tells you what tool you use. So use Clustal Omega. So that's Clustal O, number of sequences, when did you launch, when did you get the results, so on and so forth. You also get a command line version of how to run this tool. So currently, all these tools that you are running on these servers like EBI or in NCBI, these are in the background, they're actually running these computer programs which are run from command line from a Linux server, which the real command of how these tools are run actually looks something like this, right? So here you have the name of the software. There is something called in file. So after in file, you give the name of the sequences, like the FASTA file that you've input. You can tell how many CPUs you want. This shows you threads. You can tell how much RAM you want to assign. So this RAM is most likely in megabytes. So you give it 8 GB RAM. Then you give it some other parameters which tells you how do you want your result to be. So for example, you can specify whether this is a protein or a nucleic acid. If you would select nucleic acid, then this parameter value of sec type would be nucleic acid instead of protein, right? So you essentially get all kinds of information that you would require from this submission details tab. So similarly, I request you to go and look at all the tools. So for example, you have emboss cons. So this will give you a consensus sequence. So in this case, let me select DNA just to have some difference. I input this sample alignment. You know, what you can do is you can input a sample alignment that you generated from the previous tool into one of these tools as well, right? So let us say this alignment is an alignment of 4 sequences, quite long alignment. Now you can also have some more options. It can give you some name of the alignment. It can give some identity. You can get a matrix. Then you submit your job. And at the end, what you should be getting is it's a consensus sequence. So a sequence which a consensus sequence from the alignment that you have input currently. Should be happening fast.
So a small question here.
Yeah.
So you showed that there were two different trees. One was a guide tree and one was the phylogenetic tree. So what is the difference between the two?
So the guide tree, if you remember from the protocol that I discussed in the last module for multiple sequence alignment, these trees are distance-based trees in the sense that they are purely dependent on the distances. So if, for example, two sequences are exactly having the same distances from a third sequence, and each of them are having the same distance from within each other, so they might show you some guide tree which is going to have 2 nodes, like 3 nodes. But a phylogenetic tree is going to basically be a simple tree. It's going to always have sort of two out degrees, right? For each node, it's going to be a rooted tree. It's going to have more information about the evolutionary time scale. So guide tree is basically just a distance-based tree that is used to feed the alignment. So if I go to, yeah, I can just show that to you people. Change. So this, for example, in the case of muscle, this is tree 1. This would be called a guide tree. In the case of cluster W, this matrix, this one is called this is the guide tree. This one here. This is used for the final alignment. And after getting the alignment, the tree that you get after the alignment, because this one tree is only based on distances, right? But the tree that you finally get after the multiple sequence alignment, based on the consensus sequence, then you will basically look at distances with respect to the consensus sequence, and those distances will change. So you will get a different tree. So that would be a phylogenetic tree that you're looking at.
Fine, sir.
Yeah. So as you can see, you
Sir,
have.
so
Yeah.
can we say that this guide tree is based on pair-wise alignment of all the pairs available there for all the sequences which we are trying to align, do multiple
Yes.
sequence alignment, and
Yes.
okay, sir.
Yes.
And a
Guides.
phylogenetic tree is based on multiple sequence alignment.
Yes. That's
Okay,
correct.
sir. Okay, sir.
So you see, you get a single sequence, which is a guide sequence, right? Which is a consensus sequence. And here you can see wherever you have a capital letter means that all the columns, these are representing conserved columns. All right. When you have small letters, that basically means the same letter was not found in all the letters, input letters, right? So oh, I'm sorry for that. So if you look at the submission details, it doesn't give you a submission file. So if you would actually look at the file, the input file at the first location, you will see that not all the sequences would have a A. A would be the majority would be found in majority of sequences. But whenever you have a capital T or a capital A at those locations, at those columns in the alignment, all the sequences will have that letter. All right. So this is the notation that you would be following for the case of nucleotides. And whenever you have an N, the small n, this determines the locations where it was all random. So you could have 80 GCs. There was no consensus essentially. Okay. Is this clear? Basic ideas here. So please explore all
Yes.
these different tools. They tell you some information about them as well. This is so cluster omega is suitable for medium large alignments. Emboss cons is good for both protein and nucleotide alignments because it just gives you consensus score. K-align is a fast tool and it concentrates on local regions. So wherever you would have the word K, typically they would be based on k-mer based approaches. K-mers, as we know, are smaller fragments of larger strings, and therefore this is probably suitable for very large alignments. Then you have something called MAFFT, which is a multiple sequence alignment tool that uses fast Fourier transforms. How does it work? It's beyond the scope of this course, but you are free to look at it and we can ask me questions and I can explain it to you. This is also good for medium to large alignments. Then I discussed about muscle, which was an iterative alignment tool. This is good for medium alignments. This is especially good for proteins. Then you have something called MView. It transforms a sequence similarity search into a multiple sequence alignment, and it reformats this multiple sequence alignment. Basically, it's a program to view multiple sequence alignments. Then we have tcoffee, which is a progressive alignment method. It tries to essentially figure out the issues that you might have. It's an iterative alignment method as well, and it tries to figure out some issues with progressive alignments. It's typically suitable for small alignments because it gives you good, very good quality of data. So these are the different tools that you should look at specifically. Then you can go to this website, which is the Tea Coffee website. So this is like a tutorial for tea coffee. So here the idea is to understand what tea coffee is doing, right? So there are many different versions of the tea coffee tool. You can basically look at these different versions. So there is tea coffee, there is fast M coffee, there is Psy coffee, right? So just the idea for giving you this link is to just read about what these different tools are doing, how are they different, right? And for tea coffee is usually for proteins. R coffee is for RNA alignments. So there are different things, right? So now if you actually want to do the alignment, you would go to the tcoffee.org. This is how it looks like, right? And it also would be having the server. So this is exactly the same as the tea coffee server that you have here. So you can either do tea coffee from here or you can do it from this server itself. This server has other different versions of tea coffee. So for example, I can launch R coffee from here by clicking on it. Quite standard, quite straightforward, right? So you can say put a sample file and you can see this is RNA because instead of T you have use here, right? You can submit. And you would get some results hopefully very soon, right? So this was one more tool that I request you to please look at. And the other one is then going into this particular tool, multiple sequence alignment viewer. And this is a tutorial for this. Yeah.
Here you got that RNA sequences. So like, is there like place somewhere we can get this RNA sequences? Like most of the time, whatever we get on NCBI, it is like ATGC and not AUGC. So from where did they have got AUGC kind of sequence?
So I think if in such alignments, if you replace the U with the T, it typically assumes that that T is actually representing the U. So if the mRNA sequence that you get that has T's, it will be automatically taken as U's. So you don't have to worry about that.
Sir, but for here we have to like do multiple sequence alignment. So I choose like I have to do sequence alignment of mRNA. So from NCBI I will be getting the sequence which will contain only ATGC. So which one I will choose? Whether I will choose this R
Rajneesh,
copy or DNA copy? copy for DNA.
Huh. So listen to me. So what I'm saying is that typically when you are downloading things from NCBI, they would not in mRNA sequence also, they would basically be giving you something with ATGC. But because you have downloaded mRNA, you will use rCopy only. Right?
Okay, sir. Okay, okay.
Because you have downloaded mRNA sequence, na? When you will download DNA sequence, then you used the tcopy. Or when you have the amino acid sequence for that, you use tcopy. All right? Understood?
Yes, sir. Yes, yes.
All right. So now in this case, you have a nice color coding, and actually with many of these multiple sequence alignment outputs, you will have some kind of color coding, which where you will get a color specific to alignments being good. So for example, here the alignments which are good, they would have a star and would have appropriate color. So for example, here you have a star. This is all sort of green here, suggesting that the alignment probably is not so good for this one. But for this particular block, you have a series of reds. So that essentially means that you have a good score here. So in the case of R coffee, you are also taking into account how the RNA molecules fold themselves. If you remember from the tutorial that I suggested you to think about RNA, which are self-complementary, how you would use some dot matrix tools to figure those out. So anyways, just go into these different tools and explore them as well. And the third thing that I want you to do is to do this tutorial, which essentially takes an alignment. So here is an alignment of 16S rRNA. So 16S rRNA is the small subunit of the ribosome in bacteria. And this is typically having series of stretches of highly conserved nucleotides and a series of extremely variable nucleotides. So this particular tool actually or tutorial shows you how to use this MSA like multiple sequence alignment viewer. So you can actually in this kind of viewer you can see this entire 16S rRNA, which is about 1600 base pairs. You can look at the conservation here. There are different kind of colors will give you the conservation score. So probably red are locations where you have mutations, grays are locations where you don't have mutations, right? So this is example of how you would be looking at. So this gives you a legend here. Wherever you have a match, you have gray. Wherever you have a mismatch, you have these red letters. Wherever there's an insertion, you have these blue lines. Where there are gaps, there is something like this. And unaligned regions would be shown in green like that. There are some other information about these tools, how they work. And now you can, once you have loaded these things, you can actually select how to represent this data. You would want the gene names, gene symbols. Like this is more for showing like more for visual representation, what gene names, how much you would be, uh, how much column width you want to assign the gene names to. Then beyond these names and all, you can also directly go to the gene bank entries from this viewer because this tool automatically parses the names of the sequences that were used to create these alignments. Then beyond that, you can even you can go to you can fix you can zoom in to certain locations and you can look at it. So for example, we have zoomed into this particular location and you can see how the alignment looks at, right? So for example, at this particular location, the consensus sequence is a G and all the other sequences in red are actually A. So you can see, okay, these are where the mutation is happening. For example, this is a gap inserted, so you can move around in these sequences. Then you can zoom out a bit and then you can color things based on some coloring scheme. So for example, here A's are colored as green, T's are colored as red, G's are gray and C's are blue, right? So this allows you to get a quick visual representation because we are much better at parsing colors. Our eye is much better at looking at colors than at letters. Colors come to us naturally, letters are man-made. So basically, you can select these kinds of schemes for even protein sequences or similar kind of protein sequences, and you can get these kinds of color schemes. So for example, this is a color scheme that you see for proteins, right? So probably and you can also, so for example, the coloring can be based on Rasmol colors. It can be from the shape of amino acids. It can be from hydropathy of amino acids. You can also have based on some matrix scoring. So amino acid which would be more similar to each other would be of the same color. Amino acid which are slightly further away in that specific matrices would be having a different color, right? So this kinds of schemes, they allow you to look at the more conserved parts of a protein, of these proteins of interest, right? So this is the whole idea of looking at this tutorial. So once you have done these three tools, you've explored these three tools, there are these three questions that I would like you to solve. All of them are based on the same sequences, so you only have to do that once. So there are 6 different isoforms or 7 different isoforms of P53 protein. I would like you to take out their nucleotide sequences and their protein sequences and perform cluster omega alignment for them and answer the questions based on your, I mean, I want an opinionated answer here based on the alignments you see. Which one do you find more informative, the nucleotide alignments or protein alignments? Second question, I hope everybody knows what these denote, sorry. And what can you infer from the phylogenetic trees for these 7 different isoforms of P53 protein? Then I would like you to perform the multiple sequence alignment. You've already done this for cluster omega. I would like you to use MAFT and tea coffee espresso. And essentially we do tea coffee espresso and MAFT because they are much faster. And you can then compare the alignments between these three tools. And you can sort of comment on what's happened, what's the use or what differences you see. And the third thing is you perform the multiple sequence alignment of the human P53 isoforms, again, these 7 isoforms, using R coffee now, and try to see how these alignments of the mRNA using R coffee are different from cluster omega. Omega, sorry. And here also it's not cluster W, it will be cluster omega as well. Okay, is the today's assignment clear? So I'm here.
Sir,
Yeah.
I have a question. Sir, at the end, in the third question, you mentioned that mRNA we have to do. So for that, we are using R coffee. And in first question, while doing nucleotide sequences, we will be using that mRNA, right? Or something else. mRNA with cluster omega.
No, first question, you will use the nucleotide sequence. So you will use the entire gene.
Gene.
Remember, mRNA is the processed gene. So
Yes,
the
sir.
introns are not going to be there in mRNA.
Okay, sir. Okay.
Okay.
Yes. So, okay, sir.
So this is the gene sequence, and this is the mRNA sequence of the gene which is made.
Okay, sir. Okay.
I'm here. If there are any questions, please let me know. Okay.